Hi, my name is Julia Ridpath, and today we're going to be talking about bovine viral diarrhea. We're going to find out what it is, what causes it, what's its impact, and how it can be controlled. Bovine viral diarrhea is a name for a wide range and assortment of clinical presentations caused by viruses from the pestivirus genus. These viruses can cause similar clinical presentations in different hosts, so it's not just in cattle, and it's not just one virus. So what's in a name? Bovine viral diarrhea, we have multiple presentations depending on viral strain, presence of secondary pathogens, and reproductive status and age of the animal infected. We see reproductive disease that's expressed as fetal death, congenital and congenital defects, including persistent infection. We see enteric disease, the most severe of which is hemorrhagic syndrome. We see respiratory disease, principally with the infection of secondary pathogens and stress. We also see immune suppression, and this is probably the hallmark of pestivirus infections, is that the immune system is infected and directly affected. Bovine viral diarrhea viruses can infect multiple hosts, and as I said, there are multiple viral species, BVD1, BVD2, and Hobi-like viruses. If we look at the pestivirus genus, originally we just thought there was classical swine fever virus, BVD virus, and border disease virus based on the host that they were isolated from. But this method of classifying these viruses didn't work out because different viruses can infect different hosts, and the same virus can infect multiple hosts. So we went to a different method of determining whether it was BVD, classical swine fever, or BDV. And when we did that, we used genetic comparisons. And when we started doing the genetic comparisons, we found out that there were more than 3 species involved. In fact, the viruses infecting bovines actually belong to 3 different species: BVD1, BVD2, and Hobi-like viruses. Hobi-like viruses seems like kind of a funny name. It was originally based on the first names of the 2 people who isolated from fetal bovine serum that originated in Brazil, Horst and Brigitte, hence H-O-B-I, Hobi-like viruses. In some circles, Hobi-like viruses are referred to as BVDV3, but from a regulatory standpoint, I would not like to see this come into common practice, because right now we have vaccines and regulations that use the term BVDV in it. Many of the tests we now use and the vaccines we now use don't work so well with Hobi-like viruses, which would lead to some confusions with our regulations. If we look at when these virus species were first isolated, BVD1 is the oldest based on isolation. It was isolated first in 1946 in the state of New York by Dr. Fox. BVD2 was first isolated in 1987. One of the earliest isolates was by Ed DeBovie. And Hobi-like viruses, which was 2004, isolated from fetal bovine serum. To date, we have these 3 species are not equally spread around the world. BVD1 seems to be found in all continents except Antarctica. BVD2 is nearly as widespread, but thus far has not been isolated in Australia. And Hobi-like viruses seem to have the most restricted geographic distribution, being found in Asia, Europe, and South America at this point in time. The disease syndromes under the BVD tent, the immunosuppression I talked about, respiratory disease, reproductive disease, persistent infection, and mucosal disease, occur with all 3 species. The only difference between the 3 species is so far the most severe form of acute infection, termed hemorrhagic syndrome, has only been reproduced with BVD2. It has been reported with BVD1 in the field, but not reproduced under controlled conditions. Now the next slide I'm showing you shows the bovine pestivirus host range, and as we say, these viruses can infect all even-toed ungulates, not just cattle. So we have domestically sheep, goats, in free-ranging wild species, deer, elk, moose, antelope, and reindeer. These viruses can get into pigs and replicate, but they don't seem to spread once they get in as much. Variations among the bovine pestiviruses then. We have variation in biotypes. In all three species, you can have a cytopathic and a non-cytopathic biotype. The cytopathic biotype arises from the non-cytopathic biotype. The importance of these is that the cytopathic cannot establish persistent infections when it crosses the placenta. The non-cytopathic biotype can. There's also a third biotype, highly virulent. The highly virulent biotype is extremely rare, and these viruses, while being highly virulent, are also non-cytopathic. So one of the things to remember is that why the term cytopathic indicates they're cell killing. In the field, the most severe forms of disease are found with the non-cytopathic viruses. The non-cytopathic viruses predominate in nature. Most of what we're seeing out in the field is due to non-cytopathic, not cytopathic virus. Cytopathic viruses are more rare in nature. We just find them in outbreaks of mucosal disease, which occurs when a persistently infected animal is superinfected with the cytopathic virus. We'll talk a little bit more about that later, how that happens. But they're, for this reason, because they don't cause persistent infection, they're considered naturally attenuated. And for this reason, we see them quite often in modified live vaccines. So why do we have so much variation in bovine pestiviruses and all three species of these viruses? There are two mechanisms that give rise to the variation: genomic drift and another method, recombination. We'll start first talking about recombination in biotype. The genome of all three species is organized with the structural proteins on the left-hand side and the non-structural proteins on the right-hand side. At the beginning of that reading frame coding for the non-structural proteins, there's a coding frame for a protein called the NS23. It's been demonstrated by molecular biology methods that insertions into the NS3 results into a cleavage, and that cytopathic viruses have an insertion in this region, and it seems that the cleavage from the NS23 to the NS2 and the NS3 results in a change in the interaction of the virus with the immune system that allows the virus to be more easily cleared from the fetus, and so it doesn't establish persistent infection. But insertion doesn't always result in the cleavage of this protein. There are times when the insertion doesn't result in cleavage, and in some cases results in a much more virulent virus. So as I talked before about the virulent biotype, the virulent viruses have an insertion into the NS23 region, but it doesn't cause a cleavage of the protein. But somehow it changes the interaction of that protein with the immune system in a way that allows it to have a bigger effect on the immune system to, in essence, do more damage. So the practical significance for most producers of genomic insertion is the persistent infection. As I said, there's two biotypes of BVD, the non-cytopathic and the cytopathic. That's demonstrated in tissue culture, in cultured cells inside the laboratory, not in cattle. So we're not seeing more cytopathology actually means cell death, but we're not seeing more cell death in cattle with the cytopathic viruses. In fact, we see less. We see more damage with the non-cytopathic virus, principally because the immune system has a harder time eliminating it. Now, when these non-cytopathic viruses cross the placenta and infect a fetus in the first trimester of pregnancy, they're able to establish a persistent infection by means of immunotolerance. That is, the fetus, when it's infected in that first trimester, is no longer able to tell the difference between the virus and its own tissues, and from there on out replicates the virus in every tissue of its being. These animals will not die from the persistent infection directly. They may have congenital defects that lead to their death. So these animals have quite a number of different presentations. We'll talk about that later. But those animals that do survive and hit the ground are the typhoid Marys of the BVD world. They shed virus 24/7 and have a profound impact on their cohorts, because their cohorts are constantly having to fight off this virus and suffering the immunosuppressive effects of that. Early on, we thought that the fetuses infected in the final 2 trimesters of pregnancy were home free. They were born with antibodies against the virus and had cleared it. We're now beginning to understand that any fetal infection leaves damage behind, and that these animals later in life had poor reproduction values, had more treatment for disease, and had more problems with reproduction. Now, the origins of genetic drift. This virus is a single-stranded RNA virus, which means there's no proofreading function. So as the virus is replicated, with each replication, any insertion or any changes in the viral code that don't directly contribute to the death of the virus are tolerated. And because the machinery that replicates that virus is, as we say, sloppy in a way, it incorporates a lot of errors, a lot of non-lethal errors. And these incorporation of non-lethal errors contributes to the viruses forming a swarm. So as you picture a swarm of bees, all the bees are similar, but they're not all the same bee. With a swarm of viruses, all the viruses are similar, but they're not identical. Sometimes the swarm is larger depending on the conditions that it's grown under, and sometimes it's smaller, but it's always a swarm. There's always a variation. There's never just one viral strain there. Over time, the existence of swarms can lead to the development of genotypes, which we refer to as species. So genomic drift led to the establishment of the 3 species over time. At one point, there was an ancient progenitor virus that over time has mutated into these different species. It also gives rise to subgenotypes, and we'll talk a little bit about the subgenotypes that exist with each species and the significance of that in a little bit. But the practical significance of this genetic drift is changes in antigenicity. When we look at why vaccines fail, there are several reasons. One is the vaccinated animal needs time for the immune system to respond and develop a protective immune response, and this takes 10 to 14 days minimum. If the challenge occurs after the duration of protection has expired, a vaccination program will fail. That is, if you aren't vaccinating frequently enough to provide the challenge that needs to be there to protect against infection, vaccines will fail. They will also fail if the protection levels are not high enough to fight off an acute infection. So there's an animal-to-animal variation in the strength of the immune response. But the protection level may not be high enough because the virus in the vaccine is antigenically different from the challenge virus the animal's been presented in the field. And we'll talk a little bit about that now. Genomic variation creates antigenic variations. It's one of the ways the virus uses as selective pressure to preserve itself in nature. Because we saw antigenic variation between species, that is between BVD1 and BVD2, most vaccines in the United States now are bivalent, in that they include antigens from both BVD1 and BVD2, giving you better cross protection. The question we raise now is, should vaccine components be representative of field strains? Which pestivirus species and subspecies are circulating in a region? And is the antigenic difference between what's in the vaccine and the subgenotypes circulating in the region important? So our question then is, which pestivirus species causes BVD in the U.S.? To answer this question, we use genotyping of viruses that have been isolated from cattle in the U.S. We can also do comparative serology and see what cattle are responding to. Serology isn't an isolation and identification of the virus. It's asking what viruses are recognized in sera from animals in the United States. To run a good survey, you need a lot of samples, and you need them from a broad geographic area. We were lucky enough to get samples from the U.S. brucellosis testing program, which collected samples from animals originating in all of the 48 contiguous states. We got 2,000 samples from the brucellosis program within 1 year, and we tested those sera for their ability to neutralize BVD1, BVD2, or Hobi-like viruses. On the next slide, you see the areas of you see the states, and then we've divided them into geographic locations. You see that we used 1,972 samples. We got 2,000 samples in, but some of those samples just weren't adequate for testing, either because there wasn't enough of them or there were some problems with storage. So when we looked at these sera and used them to neutralize BVD1, BVD2, and Hobi-like viruses, we saw that there were a percentage of the samples that had no antibodies at all, meaning that these animals reached slaughter at 2 years of age or greater with no antibodies against any of the 3 pestivirus species. Most of the samples had antibodies that clearly neutralized BVD1 or BVD2, preferentially to anything else. That is, they had a higher titer against BVD1 or BVD2 than Hobi-like viruses. We found no sera that had a higher titer against Hobi-like viruses than BVD1 or BVD2, but we did find some sera that gave us equivocal results. And what we mean by that, we couldn't tell whether it was strongly higher against BVD1 and BVD2 or Hobi-like virus. It was slightly higher, but not high enough to be significantly important. So, but we also have to look at the level of protection in the United States. So looking at these 2,000 or 1,972 samples, we saw different levels of protection for the different viral species. You can see that about 10% had little or no protection against BVD1 or BVD2, but when you look at Hobi-like viruses, almost 20% of the animals were not protected against Hobi-like viruses. And that's having a titer of 0 to 16. We know that to protect against acute disease, you have to have a titer somewhere between 16 and 256. And a lot of the animals fell into that range. But to protect against that fetal infection requires a much higher level of antibodies present. And that can be shown in that graph that shows reduced fetal infections, titers greater than 256. And you can see that we have about 60% of the animals might be able to fight off of fetal infections with BVD1, slightly less with BVD2, between 50 and 60%. But less than 40% of the animals could fight off a fetal infection with Hobi-like viruses. This data indicates to us that while we have been vaccinating in the United States since the 1960s, we certainly have not reached the state where all animals are vaccinated and all have a protective titer against BVD. Even more than that, should Hobi-like viruses walk into the United States, less than half of our animals would be protected against Hobi-like virus infections, indicating that this is really something we want to pay attention to and to make sure that this virus does not come into the United States, because it would quickly spread throughout the national herd. The next slide talks a little about cross-protection between species, BVD1 versus BVD2 and Hobi-like viruses, mainly just showing that vaccines currently produced with BVD1 and BVD2 are not protective for infection with Hobi-like viruses. And this was done using experimental vaccines, vaccinating animals, collecting the sera, and then seeing how well they neutralize BVD1 or BVD2 or Hobi-like viruses. We also did another study in which we produced persistently infected calves and heifers. In one group, they carried calves that were exposed to BVD1, and they gave birth to PIs that were persistently infected with BVD1. Similarly, we infected heifers in the first trimester with BVD2, and we repeated that again with heifers in the first trimester with Hobi-like virus. What this slide is showing you is the titers in those animals. And what we have seen and what we believe is true is this is one of the highest challenges that animal's immune system will face, is carrying that persistently infected animal. The persistently infected fetus is shedding virus, the dam is fighting that virus off, so she tends to have quite high both T and B cell responses to those viruses, much higher than we would see against vaccination. So what I'm showing you in this slide is animals that carried a Hobi-like virus PI have much higher titers against Hobi-like virus compared to BVD1 or BVD2. Similar, those animals that carried BVD1 PIs have a much higher titer against BVD1, and those that carried BVD2 have a much higher titer against BVD2. This once again shows us the clinical significance of the antigenic differences between these species. Anagenically, they are clearly different, and cattle can tell the difference. What we also did in this study is challenged those animals with, that had been carrying a BVD1 and BVD2 fetus, waited for a second pregnancy, challenged them in the first trimester with Hobi-like virus, and even though they had titers this high, which are higher than what you would get commonly with vaccination, we were able to get the virus into the fetus. So once again, protection, while there is some cross-reactivity between the three species, infection will occur even after previous exposure, giving birth to a PI animal with a BVD virus. You can still give birth to a PI animal with Hobi-like virus. Now we're going to move on a little bit farther and talk about subgenotypes. Now, subgenotypes were first recognized by Pellerin in 1994 when he was looking at the difference between type 1 and type 2 BVD, and he noticed, he was looking at viruses from North America. He saw that the type 1s divided them in two groups, which he called 1A and 1B. Subsequently, Vilcek, looking at viruses isolated from around the world, came forward with 11 subgenotypic groups within BVD1. Subsequently, we've also looked at type 2 and found at least 3 or 4 subgenotypes in the type 2, and there appears to be at least 2, if not 3 subgenotypes among the Hobi-like viruses. So what's the practical significance of this? Well, one of the things we noticed is, as shown on the next slide, different subgenotypes predominate in different parts of the world. So there is some difference in geographic distribution. But does this have a significant meaning to in the control of this virus? Well, we did some studies looking at the impact of subgenotypes, and the results of the U.S. studies are that the BVD1b is a prevalent subgenotype within the United States, even though in our vaccines we have predominantly BVD1as. So in the United States, it's predominantly BVD1b, followed by BVD1a, followed by BVD2a, with the 1a and the 2a varying in their prevalence versus each other from year to year, but BVD1b is always more prevalent than the other two. We've seen that there's some antigenic differences among these subgenotypes. We've seen this in serological response to vaccination, once again using serum from vaccinated animals to neutralize viruses. And we've also seen vaccine escapes. So in vaccinated herds that are vaccinated with a 1A vaccine, we've seen the birth of animals persistently infected with 1B. We don't know the prevalence of this, we just know that it's been reported, and we found it a number of different times in a number of different years. We've also seen, when we talk about that persistently infected animal, remember it's immunotolerant to the virus it carries, but it can recognize and clear other viruses that aren't antigenically similar to the one it carries. And we have found that BVD1B persistently infected animals do respond to BVD1A vaccines quite well and do mount a good immune response, indicating once again there's a significant antigenic difference between these subgenotypes that is recognized by cattle. When we look at antigenic variation in BVD1 subgenotypes and look at serology following vaccination, what this next slide is showing you is the titer against 1As versus 1Bs using a 1A vaccine, looking at 6 different animals. We see that some animals, like animal number 2, give us about an equal antigenic response to both subgenotypes. But other animals show a quite different response to the subgenotypes. So if they've been vaccinated with a 1A, giving a much stronger response against the 1A than the 1B. This indicates that there, once again, is antigenic differences, but also brings to our attention that not all antigenic responses in all animals are the same. There is quite a wide range of immune response between animals. So we can also see antigenic variation among subgenotypes using classic cross-neutralization studies, that is making sera against 1A and then trying it against 1As versus 1Bs versus 1Cs, which predominate in Australia. 2As and border disease virus is shown as this slide, and as you can see, we always have the highest neutralization when we look at viruses from the same subgenotype, indicating once again that there really are significant differences between these subgenotypes. And then antigenic variation is revealed by field results. As I said, we do have cattle born persistently infected with BVD1B and herds vaccinated with BVD1A strains. And cattle persistently infected with BVD1B strains can respond to BVD1A vaccines or field strains. So there does seem to be some antigenic differences. Would vaccines work better in the United States if there were a 1B as opposed to just a 1A in there? Is a question that we would like to explore. We think perhaps we would get better cross-reactivity. Remember, once again, we're asking for a very high level of immune response to protect against persistent infection, so even a little bit of antigenic variation may increase the rate of failure with these vaccines. But now I'd like to stop talking about all the differences between these species and subgenotypes and start talking about the characteristics that they all share. Regardless of species or subgenotype, these viruses' first home in the animal is in the immune tissue. It replicates primarily in the immune tissue. It infects first, usually if with an oral nasal exposure, the immune tissues draining the upper respiratory system. Infection of immune cells leads to damage of immune cells and impaired function. It also leads to spread of the virus because as immune cells migrate out of immune tissues, they carry the virus with us. This damage to the immune cells, both in the cells that are infected and in the developing cells, leads to immunosuppression. So not only are there fewer cells there, the cells of the immune system aren't working as well as they would have before the acute infection. All bovine pestiviruses, regardless of species or subgenotype, have the ability to cross the placenta. Only the non-cytopathics cause persistent infection, but non-cytopathic of BVD1, BVD2, or Hobi-like virus all have the same ability to establish a persistent infection when they infect fetuses in the first trimester of pregnancy. All of these viruses are antigenically cross-reactive. To some extent, you can get a low-level immune response, cross-reactive response for each of the subgenotypes and the species. But as I've shown you in the slides preceding this, certainly between species, the cross-reactivity does not equate with significant cross protection. And we think this might be the case with the subgenotypes too. So, when we talk about typical bovine diarrhea, there really isn't a typical presentation. You can get reproductive disease, and that reproductive disease may be expressed as fetal death or reabsorption of the fetus. You may get congenital defects, or these animals may be born completely normal looking, or you may get persistent infection. As I said, those congenital defects may be, those animals may be completely normal looking, but they may have defects that you can't see with the eye, that are only found later when their immune system or reproductive systems come under stress. And then each in that reproductive disease is the persistent infection, that infection in the first trimester of pregnancy. But remember, any infection during pregnancy that gets to the fetus doesn't do that fetus a favor. There is always residual damage. Enteric disease, why diarrhea is in the name, this isn't the most common presentation of BVD infections. Respiratory disease, this is frequently because it is immunosuppressive and allows the secondary pathogens to have a more severe infection. These pestiviruses may also, in addition to tamping down the immune response, it may actually interact directly with other viruses to make them more virulent. It appears, we've seen this with coronaviruses, particularly with TGE. And then the big thing that all pestiviruses do is immune suppression. This is their hallmark. This is where they do the most damage. This is why they're a problem. It's because they get into the immune system. They aren't cleared from the immune system before they do damage. So, virulence and pathogenesis. This virulence of the virus affects the pathogenesis, which just makes sense when you think about it. But that isn't, strain virulence isn't the only thing that increases pathogenesis. The immune status of the host, even if you're infected with a highly virulent virus, if you have a titer of at least 1 to 16, you're still going to shed the virus, but you're not going to see all the severe clinical presentations. The maturation status of the host, we seem to get more damage of the immune system in younger animals than in older animals. We'll talk a little bit about the thymus and why that might be in a little bit. Presence of secondary pathogens is going to affect the pathogenesis you see. BVD is a big player in bovine respiratory disease complex. And some studies we've done in at the NADC doing sequential infections with bovine coronavirus and BVD, we see that both bovine coronavirus can potentiate BVD, increased pathogenesis, and BVD infections preceding bovine coronavirus infections can potentiate the pathogenesis of bovine coronavirus. And the reproductive status of the host. There is no such thing as an avirulent non-cytopathic BVD. Even though they may not cause acute disease in an adult host, non-cytopathic viruses cross the placenta. And when they cross the placenta and infect the fetus, regardless of the damage they do in the dam, they do damage in that fetus. Once again, a viral infection of a fetus never does that fetus a favor. It always leaves residual damage. Impact of infections on the developing immune system. Even as a neonate, there is impact on the immune system that they may not recover in. It's not just fetal infections that leave us with deficits in the immune system. We know there's a relationship between the age of ruminants and susceptibility to infection. And this indicates to us that there's a choreographed maturation process that occurs in the immune system. When these animals hit the ground, their immune systems are not fully developed. and they need time to fully develop, to fully fight off pathogens. Now, the thymus in young animals, in neonates, is large, and it involutes, that is, gets smaller with age. It's large in these neonates because it has a big job to do. It's actually programming or contributing to the programming of the rest of the development of the immune system. Much of that is directed by cells that are in the thymus. Stress and disease results in early involution of the thymus. That means the thymus gets smaller with stress and disease. Whereas other immune tissues like lymph nodes and the spleen, and to some extent the bone marrow, can replace damage. They regain their size. They regain the cells there that have been damaged. This doesn't seem to be the case with the thymus. Because the thymus is programmed to get smaller with age, we don't see the repair functions in the thymus that we see in other immune tissues. So this leads us to believe or to hypothesize that that early involution, or that as the thymus is damaged in a neonate, it's not able to regain those functions that it lost when the damage was done. In the next slide, I'm showing you animals 30 days past infection with a high virulent BVD. These animals were age matched. And as you can see, those animals that were not infected with the BVD have good growth, good conformation. Those that were infected with BVD, even 30 days out, just don't look as good. Where they are putting on weight, they are putting on belly fat. They don't seem to be putting on long bone growth as quickly. They don't seem to be acquiring muscle mass as quickly. A calf raiser getting these animals in would say, well, either poor nutrition or poor genetics. What they may not realize is this is the result not of a persistent infection, but of an acute infection that happened when they were neonates. So the aftermath of neonatal infection, as I'm showing you in this slide, you get destruction of thymus at 14 days post-infection. We got this data by directly weighing the thymus 14 days after infection. In non-infected animals, you can see that the weight's about 150 grams. It's about a third of that with typical virulence BVD. This is a BVD that doesn't give you a high fever or a pronounced diarrhea or a cough. This might give you a mild fever that you might not even pick up on in a feedlot situation. The enhanced virulence, the ones that give you quite high fevers and bloody diarrhea, cut that thymus even more, but you can see that 2/3 of that thymus can disappear even with what we call typical virulence BVD, what's referred to in the textbooks as a subclinical BVD infection. While you might not be getting overt clinical signs, you are certainly getting damage to that immune system, and with damage to the thymus, that thymus may not be able to come back. The next slide just shows you, in this case it was an experiment we did with Hobi-like virus, just showing you can visually see that the thymus is much smaller in animals that have been infected. And then the graph shows you that there's quite a bit of variation in animal-to-animal response. All animals show us damage, but some animals show more damage than others. Once again, as I spoke a little previously in this talk, there is some animal-to-animal variation in the immune response, either to vaccination or infection with a virus that attacks the immune system, such as BVD. The next slide shows you not only that the thymus is smaller, is that what the makeup of the thymus that is left is different. So on top, this is a program that shows you how many cells are there. So the darker red means more immune cells are present. More yellow means more just connective tissue left, the cells are gone. And what we're showing you with infection with either typical virulence BVD1, BVD2, or Hobi-like viruses, seen on the two panels on the left and on the upper right, we have, well, not only do we have a smaller thymus, we have a thymus with fewer cells left in it. And then when we go to highly virulent viruses almost entirely wipe out the cells of the thymus. So let's talk a little bit about neonatal infections again. We focus a lot on the infection of the fetus and that PI calves. But as I said, animals infected in the final 2 trimesters are exposed, not persistently infected, but may have significant immune damage that may only become recognizable later in life due to their more problems, poorer health, more problems with secondary pathogens, more problems with reproduction, more problems with handling stress. This is also what we see in neonate. So as we go from 2nd and 3rd trimesters into the neonatal stage, all these animals are vulnerable to damage with when you get infection with bovine pestiviruses. The next slide also, remember I said a little bit before that we have a lot of variation in the immune response. And when we look at an outbreak of PI animals, we'll see some PI animals that definitely look like there's something wrong with them. They have poor conformation, they have malformed skulls, the eyes are not in the right position, the coat quality's bad, they're smaller than their cohorts. But we also have some animals that appear completely normal, and actually on occasion we have had animals win blue and purple ribbons in state fairs and cattle shows and be persistently infected. You don't have to be a dumpy, ugly animal to be persistently infected. One of the interesting experiments we got into was an experiment that wasn't done on purpose. It was a university that was trying to establish a new beef herd and imported or bought a bunch of bred heifers. When they did that, they used a cattle jockey to assemble these heifers and got heifers from a number of different locations. It turns out that somewhere along the line, BVD was introduced to these bred heifers, and they had 44 persistently infected animals born into that herd. We were able to track these 44 persistently infected animals over the next year. And as you can see in that pie chart, 12% of them died as neonates, 5% of them were euthanized due to poor condition. About 41% of them developed mucosal disease. And we saw sudden death in about 15% of them. But you can see over a quarter of them look completely normal and live long enough to be sold to slaughter. And all these animals were infected with the same viral strain. So it's not just viral strain that causes the difference in the immune response and the health of these animals. It's something about their own immune systems. Each immune system responds to that persistent infection a little bit differently. What looks like a bunch of squash spiders in the graph next to that pie chart is actually the amount of variation seen in the viral swarms of these different animals. And it looks like the viral swarms are different sizes, and the size of that viral swarm, the amount of variation in the virus they're carrying, may have something to do with their health outcome in the long run. The take-home message for this is, even with the same viral strain, even being infected in the same time in pregnancy and gestational age, all PI animals won't look the same. And it's really easy to get a producer to go out there and cull those animals that look like they're persistently infected. But you can't look at an animal and say it's persistently infected. The only way to say it's persistently infected is with a test. Either you can use tests that recognize the viral genome, such as PCRs, or you can use tests that recognize the antigens, the viral antigens themselves, such as antigen capture ELISA or immunohistochemistry. But 2 tests done 2 weeks apart to establish persistent infection. You can't recognize a persistently infected animal by looking at it, by tasting it, or by smelling it. The only way is by diagnosis. So controlling the infection with bovine pestiviruses, Our overall goal is eradication. In the United States, we haven't opted to go that way. We're looking more, rather than getting rid of the virus entirely, we're looking at just limiting the losses of the infections as they occur. This is, whether this may be penny wise and pound foolish, if you don't eradicate that virus, you're going to have those costs year in, year out. So while it might be more costly to initiate an eradication program in the long run, it's going to save producers money. The tools for either eradication or limiting losses are detection. And detection primarily focuses on finding those PI animals and eliminating them. Vaccination to increase the herd resistance, and biosecurity to prevent introduction of the viruses into any production unit. The questions that producers and field veterinarians have to ask themselves: which BVD pathogen are you trying to fight off? Remember, there are 3 species, BVD1, BVD2, and Hobi-like viruses, and multiple subgenotypes. In the United States, we have primarily BVD1b, 1a, and 2a. What clinical presentation? Are you trying to prevent acute infections, or are you trying to prevent a persistent infection? The immune response needed to prevent persistent infection is much higher than that needed to prevent an acute infection. And which population are you trying to protect? Are you trying to protect the fetus, in which case you need to vaccinate the dam? Are you trying to protect the neonate, in which case vaccination of the dam leading to high titer colostrum is helpful, or you can vaccinate the neonate directly? Are you trying to protect breeding stock, or are you trying to protect feedlot stock? Once again, that immune response needs at least 2 weeks to get established. So if you're going to try to protect feedlot stock and their main challenge is happening as they are entering the feedlot, it is most effective to get the vaccines in 2 weeks before they enter the feedlot. There are also other emerging pestiviruses besides Hobi-like viruses. As we develop better and better tools to find viruses, we're finding lots more viruses out there. In the last 2 years, we've recognized atypical porcine pestivirus. This is significant because in the past we had to grow it to know it. That is, we had to be able to grow viruses in cultured cells. If we couldn't grow them in cultured cells, we had a tough time identifying that virus. We had a tough time isolating it and telling you it was there. Using next-gen sequencing techniques, they've been able to identify a brand new species of pestivirus that doesn't grow well in the tissue culture cells that we have available to us. And this is atypical porcine pestivirus. There may be bovine viruses out there like that. We just don't know yet. As we go forward, we may be finding more of these viruses. And then secondly, we also are recognizing pestiviruses in wildlife. We know that domestic pestiviruses such as BVD1, BVD2, and border disease viruses do leak into wildlife species. We have now discovered that some wildlife species have their own pestiviruses, such as giraffe in Africa and pronghorn virus in North America. So far we have no evidence of these viruses getting into domestic animals, but we really don't know whether they will go in them or not, or whether they do pose a problem. I'm going to close with a quote from Dr. Fox, who gave, when we had the 50th anniversary of the discovery of bovine viral diarrhea virus in 1996, Dr. Fox gave us this quote: "After observing and dealing with this disease for 50+ years, I conclude that BVD is an interesting, intriguing, unpredictable, and most challenging disease, which can quickly bring about financial disaster to the herd owner and apparently is going to be with us for some years to come." Dr. Fox was not only a very good scientist, he happens to be a pretty good prognosticator. We still have BVD among us, causing problems for producers. We now have tools that weren't available to Dr. Fox for control of this disease that can lead to better control and eradication. I would like to close by acknowledging a number of people at the National Animal Disease Center who contributed to the research My research career, particularly Dr. Shali Falkenberg, Dr. John Neal, Dr. Brian Vanderlei, Catherine McMullen, Patricia Federico, and Renee Lasson. We've had a very good collaboration with universities in Brazil, particularly the University of Santa Maria and Rio Grande do Sul, and from them got two excellent scientists to work in our laboratory, Dr. Fernando Bowerman and Dr. Matthias Weber. And with that, I would like to close. Thank you so much for your attention. I appreciate it.